The next topic that we are going to start with is going to be tissue processing, something that is important not only for your second year exams in your Viva, also this is important for your MCQs because you could get the order, the different fixatives and the chemicals that are used, and that is the amount of effort that gets in getting a tissue ready before you start looking at it under the microscope. So what is tissue processing? And don't worry if you feel that you'll have to learn this order as it is without me helping you with the mnemonic, that's not going to happen. Of course, I'll tell you the concept and I'll then tell you how to learn this. So you have to start with a process called fixation. I'll give you an example. For example, we are doing a skin biopsy. Okay, let's consider I'm doing a skin biopsy. So whatever things I have taken from the skin, what is the fluid that I'm going to put it inside? That is fixation. Like we did a skin biopsy and I'm putting it inside a fluid. So you will say that, ma'am, after fixation, then there is dehydration, then clearing, then impregnation. So how am I going to learn this? So please write one very famous, which I've tried to make, which can help you. You are going to fix not only the tissue, but you are going to fix the concept in your brain. You are going to fix the concept in your brain, which means what? That the first step is going to be fixation. Learn the steps first. The sounds like D. So D means there is going to be dehydration. Then fix the concept. So there is going to be clearing in your brain. In for impregnation, and brain means B for bedding and blocking. Bedding and blocking means embedding, and blocking is what is going to occur. That is what helps in getting your tissue ready. So first you have to get your tissue ready, then only you will make slides and cut it and make slides and all. That is simple, but getting this is very important. So fix the concept in your brain means there's going to be fixation, then dehydration, then there is clearing, there is impregnation, and then there is bedding and blocking. So let me start. So fixation matlab I'm going to use some fixative like formalin, glutaraldehyde, all of those. Dehydration means you have to take all the water out. You have to take all the water out of the tissue. So you can do that either with the help of alcohol or you can do that with the help of acetone. Acetone and alcohol always take all the water out. So dehydration. How do you do clearing? So to clean. Clearing means clean the tissue. Clean ke liye xylene. For clean the tissue, I will use xylene. Okay, now you say that fine, ma'am, what have you done is, for example, if this is a tissue, I will, make it all pink. Just for an example, this is a skin tissue. I have done what? I have taken out all the water out of it. I have cleaned it also, all the things. So don't you think there would be some pores that will ultimately form in the tissue because you've taken out all the water out of it. So some pores will form. Don't you feel that you need to fill those pores? Now you need to do something called impregnation, and that filling you are going to do with the help of molten wax. You are going to actually put paraffin wax into the tissue. I'll be showing you all these steps, okay? And finally, once you've put molten wax dala, that molten wax will also solidify, na? You will have bedding and blocking. I will show you this entire process of bedding and blocking. So let's start one by one. The first thing that you added, so I'm going to start this all over again. The first thing that you put your tissue in, that is going to be a fixative. So what are the different fixatives that are available? I think everyone knows the most common fixative that is used is neutral buffered formalin. The most common fixative that is used is going to be neutral buffered formalin. And how much percentage of neutral buffered formalin? 10% is what we prefer for light microscope. Whereas what do we prefer for electron microscope? 2 to 2.5% glutaraldehyde. This story I have told you a million times earlier also. 10% neutral buffered formalin for light microscope and 2 to 2.5% for glutaraldehyde for electron microscope. Now coming to the next, when do you use, what are the things I've taught you? What do you use, where do you use Carnoy's fixative? If you remember, car for car, so that is used in karyotyping. Karyotyping ke liye Carnoy's fixative, it contains what? It contains methanol to glacial acetic acid. It contains methanol and glacial acetic acid, and they ask you that what is the ratio of methanol and glacial acetic acid? It is 3:1. How will I learn this very important set of information that this is a 3:1? Because I told you to focus on Carnoy ka C and A. C is the third alphabet and A is the first alphabet. So Carnoy is C and A. So third and 1, that is going to be the ratio of Carnoy fixative that you use for karyotyping. Okay, where do you use the Bowen's fixative? If you remember the testicular chapter I have taught you, this is used for testicular biopsies. This is used for testicular biopsy, and I also told you testicular biopsy if it has been done for a case of infertility, because I want to ask you some questions. Let's see if how many of you remember this. What does Bowen's fixative contain? Bowen's fixative was containing picric acid. So I had told you that anything that contains picric acid will be very good for seeing tissues like spermatocytes, spermatogonia, primary spermatocytes, secondary spermatocytes, spermatids. Those tiny cells will be seen very well. But if someone says no, a testicular surgery was done for seminoma or some cancer or choriocarcinoma or any other tumor, then you will put it in the usual formalin only. All tissues of the body go in formalin. All cancer or any other, like breast cancer tissue will go in formalin, prostate cancer tissue will go in formalin. Similarly, testicular cancer tissue will also go in formalin only. Only if testicular biopsy has been done with a suspicion of infertility, that is when I'll put it in Bowen's fixative, because I want to see all the spermatids and all. Moving on to the next. Where do you put a bone marrow biopsy into? We can put it in Zenker's fixative, B5 fixative. Bone marrow biopsy is very good and well seen in these. Please note last one for gout. Gout has what? Gout has uric acid. Now, if you put uric acid crystals in formalin, they will dissolve out. So, I can usually prefer alcohol, or we can even prefer normal saline so that the uric acid crystals don't wash out. That is why the fixative we prefer is alcohol or normal saline. I hope the fixatives are done. You had to do the first step, that was fixation. Fix. After fixation, what are you doing? So, you will say that, ma'am, after fixation, next I had to do dehydration, that is remove all the water. How did you do it? With alcohol and acetone. Then I had to clean the tissue by adding xylene. So who is doing all of this? Do you know there's an entire machine doing all of this? So this is known as the histopathology processor. This is referred to as the histopathology processor. So if you feel that, oh my god, I will have to first pick up all the tissues, put them in a fixative matlab formalin. After that, I will pick up all the tissues, put them in alcohol. After that, I will pick up all the tissues and put them in xylene. No. After that, I will put them in impregnation matlab wax. No. Who is doing it? This machine is doing it. So this is something known as the closed histopath processor machine. This is an open. Why do I say closed and open? Because open matlab you can see all the processes like it's a live counter. You can see everything happening live in front of you. This is closed because once you put the tissue inside the machine will do all these steps and you won't be able to see them. Because you can see them open in the live, you call it an open machine. You can not see anything live out here, so it is called a closed machine. Difference or kuch nahi hai, just the view is the difference, and otherwise these are all histoprocessor machines. So first you will do fix the concept in. So remember, fixation, you've done the fixative, dehydration, that is alcohol acetone, clearing, clean, xylene, impregnation, you have added molten wax. Now what are you ready for? Now you're ready for bedding and blocking, and for bedding and blocking, I will show you this. Please note, what have you done over here? Dekho guys, look very carefully. I'll zoom in and show you. This is what is known as a cassette. In this mold or in this cassette, you are going to drop molten wax. What are you doing? See, can we all notice this was the biopsy? This was the skin tissue, the skin biopsy. Can you see that this machine is adding molten wax into it? So am I in a way doing something known as impregnation, adding molten wax into it? And after that you will say that, ma'am, we are going to do embedding and blocking. How are we going to do embedding and blocking? Please note very carefully. So firstly, what is this known as? Uske liye ye machine hai aur ye mold hai. So what is this mold known as? First know this mold, this L-shaped mold. There are two separate L's. One L is like this, one L is like this, so that I can, if I want a big, ultimately a big block, I can create the L like that. If I want a chota sa block, I can create the L like this. If I want a big one, then I can create it like this. So depending on what shape I want, this is known as the L mold. L mold ka full form hai Leuckart's mold. This is known as the Leuckart's mold. So even if you don't learn the full form, it is okay. This is just something known as the L mold. Very, very important. And this is the embedding mold. This is in which what will I do? I'm going to place the tissue. I'm going to place the skin biopsy or whichever tissue I have over here. And top of that, I am going to add all the wax. So this is going to get embedded. So this is the machine that is shown to you. This is how embedding is done. And look, finally, this is exactly how your tissue is going to look like. Can you see there's a waxy tissue over here? Can you see there's a tissue, your skin biopsy is inside wax? Look at this over here. Skin biopsy is inside wax. Can you see the skin biopsy is inside wax? So you've got something called block. Are you all clear with what was embedding? Embedding was putting the tissue in that L mold and putting wax. So you've got embedding, you bedded it, you embedded it, like you put a seed in the soil, na? Same way. I'm putting my biopsy in the wax. And after that, you're ready with the block. Everyone could see that paraffin block. everyone could visualize the paraffin block very, very carefully. What are you going to do now? You will say, ma'am, the tissue is ready. Now you just cut it and you put some blue, pink, your favorite pathology colors onto it, and let's look at it under the microscope. So what are you doing over here? You've got a blade. See, you've got a blade over here, and how with this blade you are rotating. Can you see this pathologist's hand on the side? You're going to rotate and this is going to cut and come out. You can see the gradually paraffin is getting cut. So this process is known as cutting, and cutting is done by something known as a rotary microtome. So guys, have you all seen the, in your local market around you, you must have seen tailors and how tailors work on that sewing machine. On one side they rotate the wheel and how the needle keeps working on the other side. So same way, on one side I'm rotating a wheel over here, that's rotary, and microtome means cutting. So as I rotate once, tissue is cut. Rotate second time, tissue is cut. So see, you rotate from here and this tissue will be cut. You rotate from here, the tissue will be cut. So it's a rotary microtome which you use for cutting. So you'll say, look at this, ma'am. Finally, your tissue has been cut. This is one cut slice. This is one cut slice which you put in a water bath. Why? Because that paraffin will be something like this. Aise cut hoga, zigzag, zigzag. You'll say, I want to straighten and flatten my tissue. I put it in a water bath and it gets straightened. I put it on a slide finally. This is going to be picked up on a slide. This is picked on a slide. And tell me one thing. What are the colors that you want to put on the slide? What are the colors of pathology? The colors of pathology are pink and blue. So I want to add the blue color, which is hematoxylin, the blue color, which is hematoxylin, and I want to add the pink color, that is eosin. So we all know it. The blue color is going to be hematoxylin and the pink color is eosin. So pink color to comes as a powder which I make into a liquid. Pink color ke liye nothing I want to tell you. For hematoxylin, I want to tell you because you get a question that hematoxylin actually, how did they get to know that hematoxylin is a blue color dye? Many decades ago, they used to use it for dyeing your clothes. Like clothes ka dyeing hota hai na, so blue color when they want to give to the clothes, they used to use hematoxylin. Hematoxylin used to come from Hematoxylon campesianum. Campesianum is a tree, that is the heartwood of logwood tree. Go back to the first chapter, I've taught you there as well. Heartwood of logwood tree, which is Hematoxylon campesianum, that is a tree. So I've shown you some tree barks. I've shown you some pieces and twigs of the tree from which this hematoxylin was actually extracted. So yes, we are going to do hematoxylin and eosin staining. How will you do it? By immersing it in what? Have a look at this. What is this known as? What is this container known as? Favorite question. This is known as the Copland's jar. So can you see there's a blue color solution? Matlab in this you must have put hematoxylin. You will dip your slide into it. It can be made of plastic also. It can be made up of glass also. There is no structure doesn't matter as such, whether plastic or glass. I can dip my slide into it. So if I put blue color solution, I can put hematoxylin, I can put the pink color solution, that is eosin, I can put alcohol, I can put anything. It's a jar and I can fill it with any liquid of my choice. So it is used extensively over everywhere in a path lab and a microbiome lab. So please note that this jar ka naam, this is what they're going to give you. This is a Copland's jar. And finally, your slide is ready. Can you see? The pink and blue wala slide is finally ready. And now you have to look at it under the microscope. But before you look at it under the microscope, cover the tissue, otherwise the tissue will become dry. Today, for example, your slide got stained, but you are going to look at it tomorrow morning. Your tissue will dry till then. So what are you adding? You are going to cover it with a cover slip. How do you stick to things? Kuch glue to chahiye. How do I put a cover slip on top? That medium is known as a mounting medium or a mountant. So earlier, old days mein, we used to use something called Canada balsam. Now you just need to know the name, that Canada balsam is a mounting medium. Now no one uses it. Now we use something called DPX. Now what we use is DPX. If you want to learn, it is diasterine, dibutyl phthalate, xylene. There is no need to learn the full form. Even if you learn it is DPX, it is good enough. Just know in old days we used to use Canada balsam. Now we use DPX. So you can see I've added 1 drop of DPX and the cover slip is going to stick over there and then will remain there forever. So now, before I move on to 1 or 2 questions, I want to give you a quick recap of this. What was your mnemonic? Fix the concept in your brain. So please note, fix matlab fixation, light microscopy ke liye formaldehyde, electron microscopy, glutaraldehyde. Fix the for dehydration, dehydration by alcohol acetone, concept clearing by xylene, in for impregnation adding paraffin wax, then bedding and blocking, you get that paraffin block. And how do you make the bedding? What mold? L mold, Leucart mold. Then you do the cutting. How do you do the cutting? The cutting is done by the rotary microtome. And finally you do the staining, the pink and blue staining. Once your slide is ready, how do you cover it? You're going to cover it with a mountain. In olden days you used to use Canada balsam, now you use DPX. So guys, can you tell me that help me in arranging the following histopathology processing in the correct sequence? So you will say that ma'am, the mnemonic was fix the concept in your bedding, blocking, and finally cutting and staining tha. So fix, I can see fixation, this must be 1. Dehydration, I can see that must be 2. Then clearing, I can see that must be 3. Impregnation is not mentioned here. Bedding and blocking diya hua hai, so that must be 4. So I know the order in which I want to place it. It is going to be A, D, B, C. That is what you do in the exam. So just write fix the concept in your brain and you are sorted. Let's do a match the following over here. You have to match the fixative with the condition that I have given you. So let's start. When I'm talking about Bowen's fixative, Bowen's is very commonly used for a testicular biopsy. Okay, I'll mark it over here. Then I say, where do I use Carnoy? Carnoy karyotyping. Very good. Where do I use glutaraldehyde? Glutaraldehyde is for electron microscope. I see only one electron microscope picture option given. I mark it with that. Where do I use Zenkers? Zenkers and B5 fixatives are used for bone marrow biopsies. Where do I use formalin? Formalin I kept for the end because formalin mein to every sample is sent. So whatever option is left, like a colon biopsy for that matter, breast, prostate, kidney, any biopsy would have been written, that would have been sent in formalin only, right? So yes, that becomes your fixative and the target specimen, which makes you finish the histopathology processing. Now starting with the last part of hematology, and that is going to be a quick study of these 2-3 spotters, which is right in front of you. That is a spotter known as the Neuber's chamber, and in fact now we don't call it the Neuber chamber, we call it the improved Neuber's chamber. We call it the improved Neuber's chamber. So please note, in the improved Neuber's chamber, what is the area that you count in? What are you going to count? You can count RBC, WBC, platelets. So for that matter, even fluid counting can be done. If you want to count something in the cerebrospinal fluid, like it's a case of meningitis, you want to count the number of cells. So even fluid counting can be done. For that matter, even semen analysis can be done. So a lot of fluids can basically be counted in the Neuber's chamber, but obviously my focus will be on the red blood cell, white blood cell and platelet. So please note that in this Neuber's chamber, this grid that you have over here, this one grid here and this one grid here, this is the counting area that you can see out here. So just note, where are you going to count the WBCs and the RBCs? So you can very well see the WBCs are going to be counted in the 4 big squares out here. This is what has been reserved for the WBCs. White blood cells are going to be at the peripheries. Where do you study the red blood cells? Please note the red blood cells and the platelets. So I'll show you a bigger picture. The red blood cells are going to be counted in this area. So in the central grid, firstly it will be counted in the central grid, but central grid may be not everywhere. It is counted in the peripheral 4 parts and the central 1 chota box. So red blood cell is going to be counted in a little complicated manner. The peripheral 4 chota boxes and the central most box that you have, that is the red blood cell. Repeating, the white blood cell is going to be counted at the periphery big boxes, right? The big 4 boxes have white blood cell and red blood cells are in the center. Center may be the periphery 4 chota boxes, and then you are going to have the chota center box. So red blood cell done, white blood cell done. What about the platelets? Platelets are also counted in this entire central area only. Platelets, but the entire central area. So as you can see, ye jo dot bana tha, please note the platelets are also counted in the entire central area, but to avoid overlap, they say that you are going to not count this area. These jo 2 green lines they have made, na, you're not going to count the platelets along this line and the platelets that shall be lying along this line. So these 2 2 lines of platelets are not counted, otherwise the count will come falsely elevated. So remember, platelets lying on this peripheral line and platelets lying on this peripheral line are not counted. So let's do a quick recap of this diagram. Firstly, where are WBCs counted? They are counted in the 4 big squares that are present at the periphery. And where are platelets counted? Platelets are counted everywhere in the central square, but not in the side and the upper ka 2 lines. You don't count, but in this entire central square you're gonna count. What about red blood cells? Red blood cells you count in the 4 chota squares at the periphery and 1 chota square in the center, that is a red blood cell. But for counting these cells, 2 things. What are the fluids and what are the pipettes that you use? Please note, for a red blood cell, we have a red bead wala red blood cell pipette, and for a white blood cell, we have a white bead wala white blood cell pipette. Now tell me one thing, if you see the labelings, you'll say, ma'am, one way of differentiating is red and white color. So repeating this once again, if I will see a red blood cell pipette, I will see that, okay, it will have a red bead in the center and the mouthpiece will also be red. Sab kuch red hoga. Whereas when I'm dealing with a white blood cell pipette, I will see there will be a white bead in the center and the mouthpiece will also be white. So ye bhi clear ho gaya. Now you will have to tell me that what are the markings? What if the bead colors are not given in the paper, ma'am? I will say the lower marking is 0.5 in both. Middle marking is 1 in both, but look at the final marking. In RBC, the dilution is going up to 101. In WBC, it is going only up to 11. Why? Because RBC is in millions. To be able to count it, you will have to dilute it more. WBC is in thousands, so the dilution is only till 11. So as simple as that. The bigger marking will be for the amount that's present in millions. Na? RBC is 4.5 to 5.5 millions. So you see 101 tak dilution, 100 times dilution. Over here only 10 times. It's going to be a 11 tak. So remember, 0.5, 1 and 11 are the labelings. It is WBC pipette. 0.5, 1 and 101 are the labelings. That is going to be an RBC pipette. So what will I do? You will take the blood up till here and then the blood and the fluid. That's all that you're going to do. You're going to take the blood and you're going to take a fluid. In this pipette also, you're going to take the blood and you're going to take the fluid in this pipette also. And what is the function of this bead? The bead is going to help in mixing. The bead basically helps in mixing the blood and the fluid. What fluid am I talking about? Guys, for RBC dilution, we use the Hayms and Dacey fluid. For the WBC, we use the Turk's fluid. Very famous. Turk fluid is used for WBC. Whereas for platelet, we use something which is to do with the ammonium oxalate, and that is going to be the Rees and Eckers fluid. So please remember, for red blood cells, it is going to be the Dacey and the Hayem or Hayem and Dacey fluid. So for WBC, it is going to be the Turk fluid, and for platelet, it is going to be the Rees and Ecker. How do I learn this? So dekho, platelet looks like R. So platelet ke liye to I will select the Rees and Ecker fluid. For RBC, I have learned doctor hai hum. So what is doctor hai hum? So doctor hai hum sounds like Dacey and RBC. Doctor, so Dacey, RBC, hai hum sounds like Hayem. So remember, doctor hai hum. So for Dacey and Hayem, we have RBC counting. For Rees and Ecker, so R looks like T, so Rees and Ecker ho gaya platelet, and whatever is left, that is the Turk's fluid, that is going to be for WBC counting. Simple. Doctor hai hum is going to be your mnemonic out here, and that wraps up all of your pipettes that you have along with your Newber's chamber.